Hello everyone, myself Madhu Verma, Assistant Professor, ITS College of Pharmacy, is going to present the topic SUPAC Guidelines Part 2. So, we have already discussed the components and composition changes and manufacturing site changes for SUPAC guidelines for immediate release dosage forms, for semi-solids and for modified release dosage forms. Now, for batch size, next one is batch size changes. So, we have to modify our batch size after scale up on the basis of market demand. If our market demand is very less and our supply is more, then we have to go for scale down, right. We have to decrease our production batches, and decreasing the production batches may lead to change of equipment, change of process controls, may be their change in time or temperature. So, ultimately those changes have to be evaluated and they have to be submitted to the regulatory agencies after we have already approved our product. So, batch size changes include change to a larger or smaller production batch less than 1 lakh unit scale down is not covered into batch size changes. So, if we are changing the batch size capacity to less than 1 unit, less than 1 lakh units, then it is not considered into the SUPAC level 1 change, but if the change is more than 1 lakh units, then it will be covered, and for such process we need validation, scale up validation is needed, and those need inspection also. But if the scale down is scale up or scale down is for less than 1 lakh units, then no scale up validation is needed and no inspection is needed at all. So, for batch size level 1 change, a change up to and including a factor of 10 times of pilot or bio batch where current GMP practices, standard operating procedures, controls, formulations, manufacturing etc. are all the same. Only the capacity of the batch is changing to a factor of 10, then it is included in batch size level 1 change. So, the text documentation of level 1 batch size change includes, in chemistry documentation, we have to, the product should be according to our compendial release criteria, the changes should be notified, and we have to update our batch records. The changes, the evaluation should be updated in the batch records. Then for stability data, for all the 3 formulations, immediate release, semi-solid formulations and modified release, we have to perform one long-term stability study. In vivo studies is not required for level 1 batch size changes, and for dissolution, for semi-solid dissolution testing is not required, but for immediate release and for modified release, it should be according to the official requirements for that particular drug product. And in filing documentations, we need to submit the annual report which includes the long term stability studies data. Then the next one is batch size level 2 change. It is defined as a change in batch size beyond a factor of 10 times the pilot bio batch where CGMPs, equipment, SOPs, control, formulation and manufacturing processes are the same. For level 2 batch size changes for chemistry documentations, it should conform to the release notifications of the change and it should be updated into the batch records. For stability testing, we have to perform 1 to 3 months accelerated stability study with one long term stability study. Then if significant body of documentation is available or information is available, then for immediate release and semi-solid dosage form, one batch with 3 months accelerated stability study and one long term stability study is required for immediate release and semi-solids, and for modified release we need 3 batch with long term stability studies. In vivo studies are not required, and dissolution for immediate release it should be according to the permeability and solubility requirement of the drug product. For semi-solid there should be a comparison between the current batch, the modified batch and the previous batch which is already into the market. So, there should be a comparative study for dissolution, and for modified release the dissolution should be as per the rules of extended and delayed release dissolution test for that particular documentation, for that particular drug. For filing documentations, we have to file the change being effective, matlab what the changes, what the modifications we are doing to the product. So, the anticipated changes to the quality or the characteristics of the drug product should be mentioned in CBE, changes being effective, and we have to submit the accelerated stability studies, and in annual report we have to submit the long term stability data. Then the next one is equipment changes level 1, and this equipment change level 1 is included into the manufacturing changes. Manufacturing changes is divided into two. One, either we can change the equipment, or second is we can do modification into the process or the technique which is employed to manufacture the product. So, this manufacturing changes point, it includes two things: equipment changes and changes in manufacturing process. So, it includes changes that may affect equipment and changes that may affect the manufacturing process. So, all these factors are included into the manufacturing changes. For equipment changes to level 1 of manufacturing changes is a change to automated or mechanical equipment to move ingredients, a change to alternative equipment of the same design and operating principle, or a change to different capacity equipments. So, equipment changes level 1 include the text documentation. For chemistry documentation, the changes, the product should comply to the official compendial requirements. For stability testing, we have to perform one long term stability studies. For dissolution documentation, nothing is required for semi-solid, but for immediate release and modified release, it should be according to the already approved method. Then for in vivo studies, nothing is required, and for filing documentation, we need to file our data into the annual reports, which include the long term stability study data also. Then equipment changes to level 2 of manufacturing changes means changes in equipment to a different design or different operating principle, or a change in the type of mixing equipment. So, for level 2 changes, equipment changes level 2 changes, chemistry documentation should be according to the release requirement of compendia, official compendia. We should notify the changes, and we should have updated batch records as per those changes. And if significant body of documentation or significant body of information is available, then for immediate release and semi-solid dosage form, we should perform one batch stability study with 3 months accelerated stability study data and one long term stability study. For modified release, we should perform the long term stability study for 3 batches. For dissolution documentation, for immediate release, it should be according to the permeability and solubility requirement. For semi-solid, there should be a comparative study between the two products, means the modified product and the already approved product. And for modified release, it should be according to extended and delayed release requirement of the modified release dosage form. For in vivo, no studies are required for level 2 changes, and for filing documentation for immediate release tablet, prior approval with accelerated stability studies and annual report with long term stability study data should be submitted. For semi-solids, changes being effective and annual report with long term stability data should be submitted, and for modified release, again prior approval plus annual report with accelerated stability study and long term stability study data respectively should be submitted. Then come manufacturing process changes level 1 and 2. Manufacturing process level 1 change This category includes changes such as time of mixing and speeds of operating within application and validation ranges. If the changes are within the ranges, validation ranges, then these are included into the manufacturing process level 1 change, and if the changes are beyond the validation ranges, then they are called as manufacturing process level 2 changes. So, level 2 changes category includes changes such as time of mixing and speeds of operating outside of applications and validation ranges are included in level 2 changes. Then the text documentation table for manufacturing process level 1 change includes chemistry documentation according to the release requirements, compendial release requirements of immediate release, same for semi-solids, and for modified release it should be according to the official requirements. The changes should be notified and the changes should be updated in the executed batch record also. There should be no dissolution testing for semi-solid dosage form, but for immediate release and for modified release dosage form, it should be according to the official procedures given. There is no in vivo studies requirement, and we have to file the documents in our annual report. The support document should be filed in the annual report according to the stability data. Then text documentation table for manufacturing process level 2 changes include the chemistry documentation includes release notification of the change, and it should be updated in the batch records for all the 3 categories for immediate release, semi-solids and modified release. For stability testing for immediate release, we have to perform one long-term stability studies. For semi-solids, no studies are required, but for modified release, we need one batch with 3 months accelerated stability study and one long-term stability studies. Now, then for stability of semi-solids, because stability testing is not required for semi-solid in the above row, for coming into the lower row for stability studies for semi-solid, if specific body of documentation is available, means if specific information is available for the drug product, means that is if the drug product is in the market for more than 3 years, or if we know the molecular entity of the API for more than 5 years, then that is included into the specific body of documentation. So, if data is available regarding the drug product, then for semi-solids we have to perform one batch with 3 months accelerated and one long-term stability study, and if no specific body of documentation is available with us, then we have to perform stability studies on first 3 production batches with 3 months accelerated stability studies. For dissolution, for immediate release, it should be according to the permeability and high solubility, low permeability and high solubility requirements. For semi-solids, it should be a comparative study, and for modified release, it should be according to the extended release or delayed release procedure of the dosage form. And for filing purposes, we have to file the supporting data for the changes being effective, CBE and annual report along with the stability studies data. Then manufacturing process changes, level 3 changes. This category includes change in the type of process used in a manufacture of a product, such as a change from wet granulation to direct compression of the dry product. As you already know that changing the granulation process will simply changing the compression characteristics of the product, and if compression characteristics are changed, it will lead to weight variation or drug release from the product. These both are impacted. So, ultimately these are included into level 3 changes. Now, for level 3 changes, for chemistry documentation, it should be already according to the official monograph. We should notify the changes and we should update the changes into our batch records. For semi-solids, there are no level 3 changes, right. For manufacturing process changes, level 3, there is no such change for semi-solid preparations, right. So, then for modified release, we should have the product according to the official requirements, we should notify the changes, and we should update our executed batch records. For stability testing, it's only required for modified release, and it should be 3 batches with 3-month accelerated stability studies, and first 3 batches with long-term stability studies. And then for stability of immediate release product, if specific body of documentation is already available, then we have to perform the stability study on 1 batch with 3 months accelerated stability study and 1 long-term stability study. And if no specific body of documentation is available, then 3 batches with 3-month accelerated stability studies and first 3 batches with long-term stability studies is required. Then for dissolution, for immediate release, it should be according to low permeability and high solubility studies. Then for modified release, it should be according to the already approved processes for extended and delayed release processes. For in vivo studies, for immediate release studies required, in vivo studies are required, and for level 3 changes, single dose studies are required for modified release tablets. Then for filing documentations, we have to submit the prior approval of the changes along with accelerated stability study data and annual report along with long-term stability study data. And same is for modified release doses form. We have to submit the prior approval and annual report along with accelerated stability study data and long-term stability study data respectively. Then the SUPAC conclusion. What is the advantage of following the SUPAC guidelines? First is shorter waiting times for site transfer. Then second, reducing operating overhead and maintenance expenses, more rapid implementation of process and equipment changes, improved yield and release failure investigations, more rapid implementation of batch size, it increases production of fewer unmarketable stability batches, it reduces the stability testing costs, and it reduced administrative costs for documentation of changes by the regulatory affairs departments, and if we estimate, we can save near about $51.2 million per year by following SUPAC guidelines. Now the next topic we will discuss is platform technologies. Now before discussing the topic, I'll like to discuss or give you an example what is a platform. As we all know, what is a platform? It is simply like a railway platform where each and every type of train can come, take a stop, and then it can move forward, right. So a platform, we don't have different platform for different type of trains. Like अगर कोई पैसेंजर ट्रेन आ रही है तो वो भी उसी प्लेटफॉर्म पर रुकेगी, अगर कोई सुपर फास्ट ट्रेन आ रही है, एक्सप्रेस ट्रेन आ रही है, राजधानी आ रही है, शताब्दी आ रही है, वो सारी उसी प्लेटफॉर्म पर रुकेंगी, राइट. सो दैट प्लेटफॉर्म इज अ कॉमन प्लेस फॉर ऑल टाइप्स ऑफ ट्रेन्स. That means that a simple technique is being available to various products, right. That's why it is called as a platform. It is a single place where each and every product or each and every variety is coming, using it, and then we are moving it. It is applicable to each and every different formulations or different variety of products. So defining a platform technology or platform process in pharmaceutical sciences, it is a simple single technique or a single excipient which is, or a single work practice which is applicable to a large number of products, and by doing research on one basic product, we are getting the results, we have data, and that result is being applied to other products also. So we are reducing the time of research, we are reducing the cost of the research, and it is simply, we are simply applying it to other products. Ultimately, when we are applying to the other products, our database is getting robust, it is getting strong, and we are having more confidence to apply the same technique, the same process to other products also, right. So, it is saving our time, it is saving our money. So, a platform technology or a platform process is a common or standard method or an equipment or a procedure or a work practice that may be applied to the research, development or manufacture of different products. Platform manufacturing means implementation of standard technologies, systems and work practices within manufacturing facilities and their use for the manufacture of different products, or the approach of developing a production strategy for a new drug starting from manufacturing process similar to those used by the same manufacturer to manufacture other drugs of the same type. Now, these platform technologies, these are considered a valuable tool to improve efficiency and quality in a drug product development, because we have done the research on one single product and we are applying it to the other product. So, ultimately it is giving us dividends. So, the basic idea is that a platform in combination with a risk based approach is the most systematic method to leverage prior knowledge for a given new molecule. If we are having a new molecule, we are applying our already gained information, already gained knowledge to that particular product and getting successful marketed products, right. So, this is the advantage of platform technology. Furthermore, such a platform enables a continuous improvement by adding data for every new molecule developed by the approach, and this increases the robustness of the platform. But it has often been said that access to the latest technological platforms to aid efficient drug discovery and development is limited to big pharma companies. Obviously, the big pharma companies have more money to invest into the research and development department. So, they will get the advanced techniques and they will get those techniques patented and they will apply those techniques to their own new molecules. So, ultimately only big pharma companies are being benefited by this platform technology, because they have more money to involve into that particular process. So, big companies for economic reasons want technologies which can be applied to multiple products and get those products patented. So, ultimately at the end of patent, it is made generic and at that time it will be helpful to the society. The use of platform technologies represents considerable opportunities to reduce risk to the patient safety. It provides access to platform technologies with proven performance and known safety profiles. Platform technologies applied to a particular product type, for example, monoclonal antibodies, it introduces a degree of consistency to the products under development, thereby benefiting their process and clinical development. So, overall risk associated with that particular product is reduced if we use a already verified platform technology. Now, there are some examples. For example, we know MCC, it is microcrystalline cellulose. It is commonly used as a coating agent. So, if we go through the process of coating, we know that earlier film coating in non-aqueous solvents was very popular, right. So, these non-aqueous solvents, they were creating residual solvent problems, right. We have to remove the residual solvent from the coating. Ultimately, it was creating a problem. Now, when we replace these non-aqueous solvents with aqueous solvents, then the aqueous solvent was a problem for moisture sensitive drugs. If we use this aqueous solvent coating with moisture sensitive drugs, then our drug will degrade. So, ultimately, we have to find out a platform technology. So, in this case, microcrystalline cellulose, MCC, it is used as a coating agent in aqueous coatings for moisture sensitive drugs. So, at the first time it was applied to only one drug, this microcrystalline cellulose. as an aqueous coating agent was applied to only one drug. Then later, once the process was validated, established, this process was applied to other moisture sensitive drugs also. So, in this case, our microcrystalline cellulose, it is a platform technology which is being commonly employed to other moisture sensitive drugs, and to as many as number 2 of drug products we are applying these technologies, our confidence on this technique is continuously building up that the product will be successful in the end. So, this MCC is the platform technology regarding aqueous coatings of moisture sensitive drugs. Then next one is capsules. When we go for research or clinical trials, we have very less quantity of drug, right. So, for testing that drug into a dosage form, what happens, we have to make or form a large number of tablets, maybe near about 150 tablets, to test them in a research lab or in clinical trials, right. So, if the dose or the drug available is very less, what we can do, we can make a formulation, we can prepare granules, fill it into a simple single capsule and test the single capsule. So, ultimately, if we have less amount of drug available with us during the research or clinical trial phases, then capsule can be applied to each and every category of testing, at each and every level of testing, rather than tablet, because formulating a tablet or making a tablet dosage form requires more excipients, more amount of drug ultimately. So, ultimately in this case, capsule in research and clinical trials is a platform technology. If we have more drug available, even then we can use capsules, and if we have less drug available for testing, even then we can use capsule. But in case of tablets, during these testings, if we have very less amount of drug available with us, we can't go for research or clinical trials using tablets. So, here capsule is a platform technology rather than tablet. Then next example is HPMC, hydroxypropyl methyl cellulose. As we know, all know that it is a sustained release agent for matrix tablets. So, whenever we are going for sustained release effect and preparing matrix tablet, we can blindly use HPMC as an excipient for our sustained release impact as a sustained release agent. So, whatsoever the drug we are using, if we are preparing a matrix tablets for sustained release effect, we can use HPMC. So, this HPMC is a platform technology for sustained release agents for matrix tablets. Ultimately, a most common example is we have two processors, one is Windows 10 operating system, and for Apple or iPhones we have iOS operating systems, right. So, iOS is only limited to Apple phones, right, and Windows 10 can be applicable to any type of mobile phone, right, whether it is Nokia, it can be operated on Windows, right, or Android phones, right. If we are talking of laptops, then Windows 10 and iOS systems, and if we are talking of mobile phones, then it is Android or iOS systems again. So, ultimately this Android can be applicable to any type of mobile phone, because every mobile phone is working on Android technology except Apple iPhones, right. So, these Windows 10 or our Android system, these are platform technology, because each and every mobile phone can use this technology. So, it is considered as a platform technology, whereas iOS systems is simply restricted to iPhones or MacBooks. So, it is specific technologies and this cannot be applied to other one. So, this is not a platform technology, though this can be a platform technology if Apple company give permission to use this technology on other laptops or other mobile phones. In that case, it can be a platform technology, but it is not, because they make profit with this technology by selling lesser number of products, whereas Windows 10 is applicable to each and every product. So, it is a platform technology and the other one is not one. So, this is all about platform technologies. Thank you.